Hello everyone, I'm Anas Al Ubaidi, and this is the third lecture or third session within Chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program. And the chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of drug existence and drug non-existence interaction trials. Introduction. Definition. We'll start with the definition with an introduction. As a drug existence interaction trials test how a drug interacts with other drugs, foods, or supplements. The focus is on pharmacokinetic, for instance, absorption and metabolism, and pharmacodynamic, as for instance, for example, efficacy or toxicity changes due to these interactions. Drug non-existence interaction trials examine drug behavior when other potential interacting agents are absent. This evaluates baseline pharmacokinetics and pharmacodynamics in pure conditions. Significance: improves patient safety by identifying potential adverse effects (AEs) related to drug combinations like toxicity or reduced efficacy. Helps regulatory approval by demonstrating a drug's interaction profile. Guides dosing adjustments and contraindications for complex patient populations, as for instance, for example, those on multiple medications. Example: a trial examining the interaction of a new anticoagulant drug with the statins drug existence and its clearance in the absence of enzyme inhibitors drug non-existence. Key points. Trial design. We'll start with the trial design. Effective monitoring begins with understanding the foundational design of interaction trials, as it guides the objectives, participant selection, and methods used across all trial phases. Drug existence interaction trials. These trials evaluate the investigational drug or investigational product (IP) in combination with other agents to identify pharmacokinetic or pharmacodynamic changes. And here's a summarized diagram on interaction trial designs, drug non-existence trials, and then here we have drug existence trials. Approach include combinations with commonly used medications, foods, or supplements, especially those acting on similar metabolic pathways, as for example, CYP enzymes. Objectives determine whether the combination alters efficacy or increases toxicity risks, as for example, a new anticoagulant is studied for its interaction with the statins given the common prescription of these drugs. Drug non-existence interaction trials. These trials isolate the drug under clean conditions, ensuring that participants abstain from other substances that may influence outcomes. Approach: carefully screen and exclude participants using medications or supplements and adhere to strict washout periods. Objective: establish baseline pharmacokinetic PK and pharmacodynamic PD profiles for the investigational drug or investigational product. As for example, evaluating the clearance of a novel compound in the absence of any enzyme or enzyme inhibiting drugs provides crucial data for dosing recommendations. Monitoring trial design requires vigilance to ensure participant adherence to protocols, whether managing co-administration schedules in existence trials or enforcing washout restrictions in non-existence studies. Safety monitoring. Both trial types prioritize safety monitoring to identify potential adverse events (AEs), whether linked to interactions or baseline drug behavior. Key focus areas: organ toxicity. Drugs often compete for metabolic pathways, increasing the risk of organ-specific toxicity, as for example, hepatotoxicity. Adverse event tracking, AE tracking. Close attention to AEs, adverse events, including unexpected effects arising in combination groups during existence trials. Proactive adjustments, implement dose or timing changes to mitigate immediate risks. As for example, when drug A, a statin, increases liver enzyme levels due to interaction with drug B, an antifungal medication, safety monitoring prompts dose staggering to minimize hepatic stress. For non-existence trials, monitoring may focus on baseline risks, providing a foundation to assess whether any observed toxicity in combination trials is interaction related. And here is a summarized diagram on comparing safety monitoring in drug trials, as here we have interaction-related adverse events, baseline risk assessment. As interaction-related adverse events, we have existence trials, and here baseline risk assessments, we have here non-existence trials. Data collection. Accurate and thorough data collection is essential for robust interaction studies. Monitoring needs to focus on two primary aspects, pharmacokinetics and pharmacodynamics. Here's a summarized diagram unveiling the dimensions of drug interaction studies, data collection in drug studies, pharmacodynamics, and then here centralized systems. Key pharmacokinetic metric or metrics. Cmax, the maximum concentration of a drug reaches in the bloodstream. Tmax, the time taken to reach maximum concentration. AUC, the area under the curve, indicating total drug exposure over time. Pharmacodynamic tracking. Efficacy indicators, therapeutic outcomes such as symptom reduction or biomarker improvement, toxicity signals, adverse events or adverse effects like nausea, dizziness, or organ damage. As for example, combining drug A, an antibiotic, with drug B, an enzyme inhibitor, doubles drug A's Cmax, maximum concentration, increasing toxicity risks. Monitoring systems identify this interaction early, enabling dose adjustments for safer use. Centralized systems like EDC, electronic data captures, ensure rapid and accurate recording of PK/PD data, pharmacokinetic/pharmacodynamic data, minimizing delays and errors during multi-site trials. Data analysis, careful analysis of pharmacokinetic PK and pharmacodynamic PD data reveals clinically significant interactions and informs decision-making to optimize therapies. And here's a summarized diagram optimizing therapies through data analysis, as we have from top to bottom: comparative testing, clinical relevance assessment, identify interactions, guide dosing adjustments. From PK and PD data analysis into ending up with optimized therapy decisions. Statistical methods for analysis. Comparative testing use tools like ANOVA to compare PK parameters between drug existence and drug non-existence groups. Clinical relevance assessment determine whether observed changes warrant dose adjustments or combination restrictions. As for example, a trial reveals that co-administering an antidepressant with an opioid reduces the latter's efficacy, and analysis identifies competitive enzyme inhibition as the root cause, guiding adjustments to dosing schedules. Monitoring teams must interpret these findings in the context of patient safety, ensuring any risks are thoroughly communicated to regulatory bodies and healthcare providers. Steps to effective monitoring. Step 1. Protocol adherence. Strict adherence to protocols ensures accurate data collection and participant safety. Focus area co-administration schedules, washout periods, and sample timing for PK/PD measurements. As for example, administering drug C 2 hours after drug A ensures consistent conditions across participants. And here is a summarized diagram on steps to effective monitoring in clinical trials, as here: protocol adherence, data integrity checks, adverse event reporting, and risk assessment. Adverse event reporting. Step 2, timely recording analysis and analysis of adverse events. AEs help mitigate risks during trials. As for example, increased rates of nausea in a co-therapy group trigger closer investigation into dosing frequency. Step 3, data integrity checks. Centralized oversight ensures the validity of collected samples and data at each study site, as for example, monitoring confirms plasma samples for PK analysis are properly stored at minus 80 degrees Celsius, avoiding degradation. Step 4, risk assessment. Regular risk reviews allow researchers to address emerging safety or data concerns, as for example, identifying frequent dizziness in combination groups leads to adjusted dosing intervals. Then here we have tools and techniques for monitoring. We have monitoring software, A, automates protocol compliance checks and flags deviations in real time, as for example, ETMF systems identify missed time points for PK pharmacokinetic blood draws. Here are summarized diagram on diverse tools for effective monitoring techniques. We have monitoring software, the one we started with. We have pharmacokinetic analysis, risk management strategies, and statistical tools. Pharmacokinetic analysis modeling tools quantify interactions such as clearance rate reductions or bioavailability increases. As for example, analysis shows drug B inhibits drug A metabolism, prolonging its therapeutic effect. Risk management strategies alerts across trial centers ensure emergent risks are promptly communicated. As for example, a safety alert recommends avoiding drug C in participants already using high-risk medications. Statistical tools methods like ANOVA and regression analysis highlight significant PK/PD changes, as for example, ANOVA confirms that two trial arms differ significantly in drug C concentrations due to enzyme or enzyme inhibition. Examples of successful monitoring. We have here drug interaction monitoring. Combining drug A and drug B increases toxicity adjustments to timing mitigate the risk. Data integrity, issue resolution, outliers in PK pharmacokinetic levels traced to incorrect sample storage at a specific site are resolved with targeted staff training. Adverse event supervision, higher AE rates in a co-therapy group lead to re-evaluation of dose regimens. Cross-site compliance monitoring flags, protocol deviations at one center prompting retraining for staff. And here is a summarized diagram on effective monitoring strategies in healthcare, cross-site compliance, drug interaction monitoring, data integrity, issue resolution, adverse event AE supervision. Summary tables, upcoming tables to summarize what we have covered through this lecture. We'll start here on a table we'll start with the table on definitions and significance, aspect, drug existence interaction trials, drug non-existence interaction trials. Definition for drug existence interaction trials tests how drug interact with other drugs, food, or supplements, while definition for drug non-existence interaction trials examines drug behavior in the absence of interacting agent. We have the focus and another aspect, significance between both, key goal and example for each. Table on key components of monitoring. Component description, how it's ensured and example. So components, we have trial design, safety monitoring, data collection, analysis, and compliance. Table on steps to effective monitoring. Step, objective, activity, how it contributes, an example. Steps, we have protocol adherence, adverse event reporting, data integrity checks, risk assessment, and feedback loop. Table on tools and techniques for monitoring. Tool/technique, purpose, how it works, benefit, example. And the tools /techniques, monitoring software, pharmacokinetic analysis, risk management strategies, real-time data monitoring, and then statistical techniques. We have examples, table for examples, or a table on examples of successful monitoring, scenario, challenge faced, monitoring action, outcome achieved. Scenarios, we have drug interaction, drug outlier detection, adverse event pattern, cross-site compliance, and then risk mitigation. Application table, scenarios. For each scenario, key principle applied, key actions, timeline, and what to watch out for. Scenarios, we have tracking interactions between a new anticoagulant statins and drug interaction trials. We have testing drug clearance in isolated conditions in the absence of external factors. We have here identifying increased hepatotoxicity due to enzyme competition between two combined medications. Ensuring drug administration sequence in accordance with clinical protocol timing, flagging anomalous drug concentration spikes in one participant group, adjusting safety thresholds for a trial due to emergent toxicity patterns, comparing drug A and drug B to determine interaction effects on therapeutic consistency, and here we have investigating high risk rates of nausea in participants receiving combined drug therapies. Continuing with scenarios, here we have real-time adjustment to participant dosing strategies when cross-site trends reveal interaction risks. We have using ANOVA to evaluate efficacy of non-interacting agents compared to those combined with other drugs. And then we have the scenario of remote oversight of PK pharmacokinetic sample collection processes to ensure integrity across international trial locations. Continuing with scenarios still. Real-time adjustment to participant dosing strategies when cross-site trends reveal interactions risks, using ANOVA to evaluate efficacy of non-interacting agents compared to those combined with other drugs. And then we have remote oversight of PK pharmacokinetic sample collection processes to ensure integrity across international trial locations. Next, we will be revising what we have covered through some MCQs or multiple choice questions. Question 1. What is the primary difference between drug existence and drug non-existence interaction trials? A. Drug existence trials test drugs in isolation, while drug non-existence trials evaluate drug combinations. B. Drug existence trials focus on interactions with other agents, while drug non-existence trials assess drug behavior without such interactions. C. Drug existence trials only track adverse events, while drug non-existence trials focus on PK parameters. Or D. Drug non-existence trials monitor long-term safety, unlike drug existence trials. As not drug non-existence trials monitor long-term safety, unlike the drug existence trials. So the answer would be B here, definitely, as the question, the difference between drug existence and drug non-existence interaction trials, as the answer would be B, as B is the most accurate correct answer, as drug existence trials focus on interactions with other agents, while drug non-existence trials assess drug behavior without such interactions. Question 2. Which parameter is tracked during pharmacokinetic analysis in drug interaction trials? A. Therapeutic efficacy scores, AUC area under the curve values, recruitment timelines, or consent from completion rate. Consent from completion rate. So, which parameter is tracked during pharmacokinetic analysis and drug interaction trials? So it is area under the curve values. B, as B is the correct answer. Pharmacokinetic analysis evaluates measurements like AUC, the area under the curve, which reflects total drug exposure. The other options either refer to non-PK parameters or non-relevant administrative tasks. Question 3. Why is safety monitoring critical in drug existence interaction trials? A. To ensure compliance with trial budgets. B. To identify adverse effects caused by drug combinations such as toxicity. C. To track participant recruitment data. Or D. To verify proper PK pharmacokinetic sample refrigeration. And definitely it's B, as to identify adverse effects caused by drug combinations such as toxicity. As here, why is safety monitoring critical in drug existence critical trials? As to identify adverse effects. A is caused by drug combinations such as toxicity. So B is the answer. As safety monitoring identifies and mitigates potential adverse effects or toxicity associated with the drug associated with drug combinations, which is fundamental in drug existence interaction trials. Question 4. What is key feature of risk management in drug interaction trials? A. Eliminating high-risk participants from the study. B. Developing a risk communication plan to alert stakeholders about safety findings. C. Avoiding statistical tools and data analysis. Or D. Delaying safety reporting for resource optimization. As which is the fundamental in drug resistance interaction trials would be B. Developing as a question 4. Sorry, what is the key feature of risk management in the drug interaction trials? As the explanation here, safety monitoring identifies and mitigates potential adverse effects, AEs, or toxicity associated with drug combinations, which is fundamental in drug existence interaction trials. This is explanation as we explained for the answer B for question 3. But here, what is the key feature of risk management in drug interaction trials? The key feature of risk management, as would be B, as the key feature of risk management, as B, developing a risk communication plan to alert stakeholders about safety findings. As a risk communication plan ensures that urgent safety updates are swiftly communicated to all relevant parties, enhancing trial safety and efficiency. Question 5. When unexpected spikes in drug concentrations are detected during a trial, what should be the first monitoring action? A. Conduct immediate root cause analysis. B. Halt the entire study until further review. C. Increase participant recruitment to offset errors. Or D. Remove all high-risk medication from the trial. So, when unexpected spikes in drug concentrations are detected during a trial, what should be done as a first step? Or first, should be the first monitoring action? A. As the immediate step would be conducting immediate root cause analysis. A is the answer. Whenever abnormal results are identified, such as unexpected drug concentration spikes, a root cause analysis is essential to determine the underlying issue and implement correction strategies promptly. And here we come to end of this lecture. Thank you very much and goodbye.